I kindly request the dignitaries to kindly join the audience. We are pleased to extend a formal invitation to Dr. Ashwini Godbole as the speaker for the third scientific session. This session will be presided over by Chairperson Dr. Pawan Kumar and Dr. Srinivasa Rao sir.
To begin with a small introduction of Ma'am, Dr. Ashwini Godbole is a distinguished researcher in neurobiology and Ayurveda-based cognitive health, currently serving as the principal investigator for Ayurveda neurobiology research at Transdisciplinary University. With extensive training in molecular biology, cell biology, and mitochondrial research, her works bridges classical Ayurvedic wisdom with contemporary scientific methods. Her research focuses on Medhya Rasayana, healthy aging, neurodegeneration, supported by major national and international grants. A recipient of the prestigious fellowships from DBT, DST, SCRB, and international scientific bodies, she has published widely in peer-reviewed journals and actively contributes to curriculum development and global scientific forums.
Namaste. I begin with thanking the organizers, Jaikarji, and whole team, and thanks to all of you to be here, and it is nice, it's a pleasure to be with so many Ayurvedic Vaidyas and showing them something which is now become my passion. And I consider that because Ayurveda has that nature, very vibrant, at the same time very deep. So that's the reason a scientist like me, who's trained in modern biology, cell biology and biophysics, can keep on working on one single concept from Ayurveda forever. And as I said earlier also, I'm so sure that such kind of concepts and such kind of practices exist in every Indian system of medicine. That is Yoga, Unani, Siddha, Swaripa, all of these. One researcher like me can work on a very little what Ayurveda or Indian system of medicine has said. And if all of us join together, maybe we can do much more and offer it to the world. That will be unique offering from India. So moving forward, this is one step for the dream which Pawanji showed us. Such a big dream. You can be actually someone who can decide national or global policies for traditional medicine. Excellent to see that we do have that kind of human resource and that kind of knowledge base which can take us there. And then after that, Bala showed you that how we can use what's happening. And then Bala showed us that how we can look at the data which is getting generated and using the Ayurveda as a base, and then data getting generated, and we can then make some tools like AI, using AI, and use them in day-to-day practice. I'm going to look at very much smaller one concept from all of this and going to talk to you about it. And why did I choose doing this? Because over time, while working in a university called as Transdisciplinary University in Bangalore and talking to so many Ayurvedic doctors and even the folk healers, I learned that there is a lot to offer from these systems for health span, remaining healthy. And we talk so much about illness. Everybody goes to the doctor only if they fall sick and if there is some health problem. But can we talk about wellness? More we talk about wellness, more wellness will be generated and more illness will automatically be reduced. And I thought the training which I have got in biology helps me to do that, because then I can show at the cell, I can see first and show then that at the molecular and the cellular level, health and disease is in continuum. So if you remain towards the health side, the disease won't happen. And that's so very obvious from the biological point of view. If that such a obvious information, if I can put forth, then and how Ayurveda allows us to remain in the health side and not fall sick, then I think we will be will be very happy and content, and we will live a meaningful life. I think apart from pharma industry, nobody will be unhappy with this. So that's what is the thing. So I hope I can convince you, especially the young Ayurvedic Vaidya or Ayurveda students, that there is so much of scope in for us to do a very positive and promotive, health promotive kind of work in this field. So that's what I would like to begin. The whole point of Ayurveda is swasthasya swastha rakshanam. The first thing is that, and if at all, then after doing that also, if there is disease, then we can take care of. That's why Ayurveda, that's why I'm convinced that a science which is already saying this as a motto will definitely achieve this, because that's what the science is meant for. So that's why Ayurveda is a way for healthy, happy, positive. I'm not just saying preventive. Prevent disease is a very minimal kind of expectation. Of course it will prevent disease, but we should live a much happy and healthy life and a very meaningful life. And because Ayurveda is so comprehensive, and when I keep saying Ayurveda, I mean to say the Indian system of medicine is Ayush, is so comprehensive that it will allow us to actually achieve this very much easily than what we expect. So that's what I would like to convince you in next half an hour or so. So my area, means my group's area of work, is about aging and the nervous system. So healthy aging and cognitive wellness, or nervous system wellness. So why this topic? Because as you all know, that we are living longer, the population is living longer globally, but the health span has not increased as much as the lifespan has. What do I mean by that? Is that the people are not living healthily, especially last couple of decades of the life. So that means aging is a risk factor for many diseases which are coming. If the aging is healthy, then the diseases won't happen. And if we can understand that from the Ayurvedic point of view and from the life science point of view, maybe we can find some very easy solutions which we can do. So this is a dialogue between Shastra, research, the clinics and the communities. So it's not one way dialogue, it keeps going back and forth. As we have to keep saying that we sometimes listen to Shastra, design an experiment, look at the practice, whether it is used. Why am I doing this? Because it should be practiced. Then look at the communities. Will they accept it? Will it be done? And again go back to my lab and do some research. And this circle has to go on. The dialogue cannot stop for a long time. So again in a different way, and we have to do this merging of Shastra, practice of the science, whichever Shastra you are interested in, and the science very seamlessly. We have give and take in all the domains. And this is what TDU provides us. TDU, as I came to TDU, as when it was the FRLST avatar, we were majorly known for our conservation of medicinal plants and local health traditions. Then the science part of it and the clinic part of it came. But I have seen this is one place, and there are some other places, and there should be many more, where science and Shastra is at same level. There is respect from Shastra to science, from science to Shastra. There is a lot of passion to find what the kind of research which I will show you, to do that kind of research. And we also have the infrastructure to do it, where the people, the infrastructure exists where the human resource exists, meaning where the Vaidyas are, where the scientists are, and there is the basic infrastructure which we can utilize to do this kind of research. So such ecosystems have to be built, and that's what I think very useful for a person like me who wants to do Ayurveda biology. So what do? We start with we start with the concepts of Ayurveda, and then look at the practice of Ayurveda, then design our experiments, whether we need to do a clinical study or whether we want to do the lab study, which way we should go and what kind of lab study, how deep, what kind of experiments and so on. So I'll just show you one example. As I told you that we look at cognition being three things at least. What are those three things when we say cognition? It is MAC, that is memory, attention and concentration. That's what is known in the modern cognitive neuroscience, that these three are the minimally required things for when we are measuring cognition. And a very similar or a very parallel kind of a concept of pragya, which we first, when I started around 8 years ago, I went to Vaidyananal and I was with him in his clinic for 3 days, and I kept asking him questions once in a while. Maybe I was irritating him also a little bit, but I kept asking him and he was nice enough to keep answering those questions. And after that, and after reading his book Man and Ayurveda, which is in Marathi, I learned this. At least I could get this one slide after so much of this thing, that there are three faculties: Buddhi, Medha and Smriti. And Smriti is easy to understand, memory. Buddhi is something which will allow us to take whatever we want and leave rest. That means we will take the signal and noise will be lost. So there was some song going on, there is some light, but we do see them, but we don't pay attention to them. And that's what according to Ayurveda, manas, that is the mind, allows us to do these things. And Medha, which is the in-between entity, which is whatever you are gathering, whatever you are grasping, whether to make it into information, memory or not, how important, you assign importance to it, and then that allows you to do that. And this faculty can go up by various things which we do in our Indian system of healthcare, I would say, including meditation, yoga, so many things. Even in the modern also, there are certain things which can be useful for us to make this decision, what is good and what is not. And so that's what at the end he was also saying, that whether AI will replace that. No, it will not, because AI doesn't have medha. It will have buddhi, it can have smriti, but medha is only the human quality. So we have to see that this is what it is. So then medha again also decides that I have certain memory, right? I have memory about Chennai, saying that Chennai is very humid and hot. But I will use certain power of mine, and then when I see, and I say that no, this is January maybe. So we know how to deal with the memory which we have. Also about people, we have certain memory. Anything, we can have memory about anything, but our behavior is not exactly the memory which we have. It is permutation and combination of many other things which we assign to the memory, and the behavior is governed by that. And that faculty is again medha, which governs that behavior. That's why maybe Ayurveda gives a lot of importance on Medhya Rasayana and not so much about the Buddhi Vardak or Smriti Vardak. There are Smriti Vardaks and Buddhi Vardak, but Medhya Rasayana is a very unique concept which exists in Ayurveda. So it's not only about health. So you will say that if you keep on doing only health, then we know that when we say that Ayurvedic practice is very subjective, and when we are trying to do research in Ayurveda practice, how will we do it? We don't use, there are no, like every Vaidya can give a different outcome, as Bala was also showing something like Prakriti. But then if we think how we should do research, so I just wanted this slide to be put for the students mainly, that we can have algorithms. We can have algorithms. If you take one type of disease to all people, like say Parkinson's, this is for Parkinson's and stroke. If you take if a person who has Parkinson's goes to Ayurvedic doctor, they will follow an algorithm. Maybe the choice of drug can be different, the choice of medicine can be different, but the algorithm will remain same. That means the pattern remains same. So that's what we should be able to take out from all this information of which we are gathering from the practice and the experiments, and that's when it will help to develop newer and newer tools to look at these things. And we do get best results. Even if we take 10 Vaidya's advice, every patient which is put into this graph has been treated differently. Right? The result is very good in most of the cases, and we know in which cases the results won't come like that. That is also known. So if we understand, use this algorithm pattern, then I think we will learn much more than looking at the exact details of the pattern. So what do we do? So we do not do any clinical research in the clinic. We do community-based clinical research, because what we want to achieve is wellness, right? So the person won't come to the clinic. If person is hell and hearty, good for them. If they are there, then they won't come to the clinic. But we know that if something is rasayana, something which we need to take care, that in future it should not happen, we need to work on it today. If we don't want it to happen, or if we want to remain healthy for a long time, we need to start working every day from today. So, or maybe better if yesterday. So we need to see what are these patterns. So for cognitive health, we go to the community, we go and talk to them, we do cognitive tests, which are simple paper pencil tests, we do blood tests, and we also do the sleep, anxiety, depression, because these are very soft issues but very, very important for as far as brain health is concerned. We do blood test, of course, before and after. We do MRI, which is functional MRI we do. And why I am showing you all this? Because I think the results which we will show you, I will show you, are very about one formulation of Ayurveda, and how much can we learn from one formulation of Ayurveda. That's what I want to convey. We of course use Prakriti and Agni as tools for diagnosis. Agni is done before and after, Prakriti of course only once. And what do we look at? We look at only one formulation, because I went to 10 Vaidya. I asked them, if I want better cognition, what would you give? Anybody who wants better cognition, what would you give? So how many Vaidyas here? All Vaidyas, right? Many Vaidyas. And even Siddha has some similar formulation, little different composition. But everybody said Brahmikrita. Everybody said, they might have said something more, but definitely Brahmikrita, one of the formulation. So we started this study with Brahmikrita. Then the control was plain ghee, because in case of Ayurveda, base is not inert. Base is also an active player in the movie. So this, only Brahmi, only ghee and placebo. So that's the thing which we did. And we used I call it a wellness dose. That means normally if there is a memory decline, Ayurvedic Vaidya will give it twice a day, 6 grams, but we use only once a day. And we want to keep it wellness intervention. So we don't tell people that a lot of do's and don'ts and all that. We tell them, take one in the morning, Brahmi ghee or Brahmi extract, and after that you can carry on with your own routine. So that's how, I think that's another thing which we need to do when we are talking about wellness. If we give a long protocol, maybe it's difficult to do it every day, and we want people to do it every day. So that's the reason we did this way. So now let's see whether you can be a participant in our trial or not. So our study or not this is the question. So everybody, little bit difference. So everybody please look at this picture. I'm going to ask a question about this. Yeah, 10 seconds. Which ones of these were in the first picture? Which of these pictures were in the first picture? Huh? See? E? Yeah. B and E. See? Looks so simple, right? Looks so simple, but it is not. And it is. So these are the kind of simple things which we do during our cognitive tests. So the person also can enjoy. There will be a word list, there will be a story, which we will narrate, and after 20 minutes we will ask, so what was there in that story? So it's very, so we have to do these simple things, and that gives us a lot of information about the data is quite dense. But what happens is that we also give such kind of things. Like, what time is it? In the clock, 2:30. So if we just ask people to draw a clock, first of all, this generation is so used to of looking at digital time, but draw a clock, place the numbers and draw 2:30 time in that clock. Normally, most of the people can do it. But as people start aging, 50 plus, the speed reduces. It will take longer, and then if the things are not as good as they should be, then that's what happens in mild cognitive impairment or early Alzheimer's disease. People start misplacing the numbers and the time might get flipped. It's kind of flipped. So these kind of things, and in AD, if they have Alzheimer's disease or dementia, it's worse. But we have seen that we can increase the speed when the person is normal. They can do the things faster if they are taking on Brahmi or they are on Brahmi Grita. If they are in MCI category, they definitely switch very close to normal. So those are the places where we have a lot of scope. Yeah, so what happens then? So if we have, it was very nice to see that we had people in two groups, 25 to 55, with whom we call younger adults, and 55 to 85, who we call older adults. So we saw, so I was coming, however I tried to be, we thought that Brahmi Grita is a better formulation, it's given in Ayurveda, but we wanted to do an unbiased study, so it was an assessor blinded study. So assessor blinded means, it's very, why I want to bring this point is that it's very difficult to do double-blinded placebo-controlled trials many times in our Indian system of medicine. Imagine something like meditation and yoga. What blinding will you do? Is it, or even for ghee. A person is eating ghee, that person will know that person is eating ghee, right? We all Indians know the taste of many, many things. So, I don't think we need to follow the protocols of double-blinded, placebo-controlled trial and all that, but we need to be unbiased. So the assessor, the person who's doing the cognitive test, was blinded for what the person is taking, the individual whose participant is taking. So we saw that the earlier age groups, we thought Brahmi is not working because the cognition was already very high and the Brahmi effect was not, that won't be. But believe me that the younger population had more of sleep and stress problems, unfortunately, than the older population. And plain Brahmi, which is Bacomine, actually helped them to get better quality of sleep and get less of stress. So that's what we saw. But you can see on the in the rightmost column, the aging population, the 55 and above, actually showed lot of benefits of Brahmi as ghee, in the ghee formulation, as with the base, ghee as base. And that's what we have been again and again seeing. What is given in Ayurveda? Ghee is medhya, first of all. Secondly, it is given that any degenerative condition, the fat base will be useful. And that's a very unique proposition from Indian system of medicine. Siddha, Ayurveda, and I don't know much about Unani, whether Unani offers such, but I definitely know Siddha and Ayurveda has this. And we can see that same thing we can see in this results of clinical trials. Now one can say that we need more data. Denser the data, better it is, right? So from MRI, what can we see? We have two types of cells in the brain. Anybody knows what two types of cells we have in the brain? We have two types of cells in the brain. One is neurons. Other? Anybody knows? I just want at least you to get thinking. Other is something called as glia, which supports neurons for its function. It's called glia, the word comes from glue, and then it supports the neurons to function well. But what we see is, neurons are okay, but as the person is aging, the glia starts giving up. There is more inflammation even in the brain, and the choline level, in the first graph you see, the choline level increases, which is indicating that the glia, there is some problem in the brain, mostly with glia, and myoinositol also increases. We saw with Brahmi ghee, just 3 months of Brahmi Ghita, the choline came down. The stress in the brain came down. So that's what we have been seeing repeatedly, that at multiple levels we can see that the Brahmi Ghita is being useful. Same thing we see with functional MRI, there are networks, and we see that with Brahmi there is advantage in 2 networks. The 2 networks become more robust if the person is taking Brahmi ghee. So, but then the question comes that the ghee will increase my cholesterol, triglycerides, but that doesn't happen with Brahmi Ghita. Brahmi Ghita does not increase unfavorably, does not affect the lipid profile unfavorably. And we have more than 200 people taking Brahmi Ghita, so we know that this is consistently we have seen. Either it will keep it normal, same, or it will reduce. And so that's what we have been seeing, the cholesterol and the triglycerides. So that's what we have been seeing repeatedly. And one other interesting fact. So when you do research with open mind, you see once you collect enough amount of data, you start seeing lot of patterns. One of such interesting pattern which we saw from our research was this, and we have published this already, that people who have diabetes, even if it is under control, even if they are on medication, their cognition is lesser than people who do not have diabetes. So a person who has diabetes, who is 40 years, I'm just giving you just to drive the point, I'm giving you these numbers. Who is 40 years, the cognition will be like person of 60 years. So that's how glucose metabolism affects your systems in the body. So now we have talked about cognition. I showed you that they can do puzzles better, they can remember the stories better if they are taking ghee. But how do I know the anti-aging effect of it? Because there are more and more number of data that results which show that if you are aging healthily, then the cognition also is good, the movements are good, the nervous system is good. That's the reason why we then go to the lab, which is my forte. I am a biologist, so going back to the lab is always very nice, and we see that there are levels at multiple levels you see the similar effects. So what do we do instead of, so if I have to look at the anti-aging effect of Brahmi Ghita, I have to do 70 years experiment, right, with human beings. At least 70 years I need to follow. Who will give me the grant for doing the experiment for 70 years? And how many people will I be able to follow for 70 years? I cannot do that. But I have my friends in the lab, which are flies, worms, rats, mice. They will follow what we have to tell them, and their lifespan is it's very short. So we can actually very intelligently look at the same formulations which we have seen the clinical effects, and then use them in the lab to look at the deeper molecular and cellular understanding. What kind of questions can we ask? So for brain, Ayurveda says, for cognition, Ayurveda says Brahmi or Mandukaparni. For peripheral nervous system, it says, first it says Brahmi or Mandukaparni in ghee. The ghee is the base. Otherwise, for the motor health or the peripheral nervous system, it says Bala with milk and oil. So the base is different, the herb is different. Now all the permutation combinations, if we have to do, we have to set many experiments and do it, and that's what we do. And there is another herb called as Vacha, Acorus calamus, which is also good for brain. But in case like stroke, epilepsy, which are functional disorders. So Ayurveda has a very clear-cut understanding. Something which is degenerative, you give the top 3. That means Brahmi, Mandukaparni, Bala, with the base of either ghee or oil and milk. But something which is functional disorder, that time you give a sharp drug like Vacha. So it's such a very, very beautiful understanding. And if we learn the biology or the biochemistry and biophysics behind it, I think we can propose these as wellness solutions to the world. So that's what we are trying to do. And the other medicines also we are studying, but other herbs also we are studying, which are good for, supposed to be doing particular job in the nervous system. So what do I use in the lab? We don't even use mice and rats. They are also too big. We use an organism called Caenorhabditis elegans, which lives only 21 days. So my experiment is over in 21 days, unlike 70 years, not 70 years. So why we use this organism? You can see the Nobel Prize for the first aging pathway and aging gene was found in this organism. And the way the worm ages, it looks very different, but the genes which are responsible for aging and the nervous system function of the worm are very similar to what we have in human beings. 60 to 70% genome similarity at the functional level. That means 60 to 70% of the genes which function in a particular way in the worm, similarly they function in us. So we know that, like say Parkinson's, we know dopaminergic neuron, dopamine is responsible for movement. I will show you the data further. In the worms also that's the same. The worm has 1,000 cells. 1,000 cell makes a worm. 309 are neurons. And every neuron, you can see the bottom figure, every neuron is known in the worm, how it connects. So we have, in short, what am I trying to say is we have lot of information about the system. Unlike we are a very complicated organism. We cannot be understood very easily, but we can understand these small organisms easily, and also that I can do experiments which Ayurveda says. What does Ayurveda say? Rasayana treatment in middle age or young age will give you benefits in the old age. Now I can feed only the tiny little worm, and I can stop feeding them Brahmigrita later and see at the old age. Or I can feed the worm in the middle age and look at the old age. So various types of experiments can be done and lot of information can be gathered. And we do these kind of experiments. We have the central understanding from biology that there is a lot of molecular damage happening during aging. There is energy dysregulation happening during aging. That's why the glial, which I explained to you, glia give up. That's why there can be neurodegeneration. And that we are looking at Shrota Shodak, the sharp pungent drugs or vegetables and fruits which are given in Ayurveda as Agni Vardhak or Shrota Shodak. Can we use that? And a very unique concept from Ayurveda and biology says that if your system is very well adapted, if the adaptation is good, then all these things which are happening during aging will be slowed down, will not affect you in bad way. So Ayurveda has the concept of poshana, and I think that's where the milk and the oil and ghee falls under, that if we are consuming certain types of material continuously for a long time, then that makes our system resilient. This word resilient is very important because that means that you are not correcting the stress. It's not anti-stress. You will not feel the stress. Do you see the difference between you will not feel the stress? Even if the same thing is there, you will not feel it. The stress will be there will be a lot of work, but you will be able to handle it much more efficiently and you will not feel stress. That's why I need to take something, I need to have a cup of coffee to relieve my stress. That's not the thing. You will not feel the stress. So that is what is resilience, and Ayurveda offers these unique propositions that you will build resilience. You will be much healthier that way. So this is what we see, the same Brahmi Gruta which we give to the people from the community, we fed the worms. They don't have a choice, they are in the plates and they eat whatever we want them to eat, so most of the time. So we see that the same Brahmi Gruta which improved memory and cognition in human being, improved the lifespan of the worm. And not very little, quite a lot. 30-40% increase in lifespan. The ghee, plain ghee also did half of the job. So the ghee is very important, it's an active player. Brahmi and other herbs make it better. And we could see that happens even in case of Ksheerabala, another formulation. We can see that they are not just living longer, they are also healthier, they can move better for longer time, they can do all the activities for the longer time. That means their health span is much better, and while the lifespan is increasing. And we saw the similar kind of thing in mice. So you will see that the worms are much lower organisms, and we want to see the first results in the worm-like system, then take it to the mammalian system, and we see that the 2 months old mice versus 6 months old mice and the 12 months old mice, and they are So 12 months old is really old, 2 months old is very young, and 6 months is a middle-aged mouse. So you can see that if you make them hang on the wire, the 2 months old will not have any problem. They will hang for a very long time. They will climb the wire and get off also. But as they grow old, 12 months old do have a problem. And in case of 6 months and 12 months, in both the cases we see lot of benefit. That means in absence of a disease, the mouse is not diseased. It is the normal mouse. But even in absence of the disease, Ksheerabala in this case is helping to make the health better, make the mouse more resilient. So this is what we see, and we see this at the, as Bala was saying, we see it at the behavior or the phenotype level. We also see it at the genotype level. That means the muscle building genes are higher, muscle degenerating genes are lower in these treatments. That's what I'm trying to make a point, is that it is makes the system more resilient, makes the system more robust. So same thing, I'll go fast. You can see the brain of the 12-month-old mouse looks like this. On the right hand side, what you see, you will see a lot of gaps. You are seeing a lot of gaps in the brain tissue, but with Ksheerabala, we don't see like that. So you can see the difference between this one, the gaps on the right hand panel, and with Ksheerabala it does not happen like that. And we have been seeing this. The base is also very active. The base of Ksheerabala also does the job almost equally well. So these are certain points which we have learnt over time, and we can do that, go on doing that. Now we are looking at aging, then we look at the disease, and if the worm gets Parkinson's disease like human beings can get, we can also look at that. So normal worm will look like that, move like that. The worm which has Parkinson's disease, again there is lack the dopaminergic neurons, same neurons which degenerate in a Parkinson's patient, same neurons if degenerated in the worm. You can see that these are on the left hand side on the top, what you see are the dopaminergic neurons. If they degenerate, this is what happens in the worm. Normally moving worm, if the dopaminergic neurons degenerate, they have problem in movement. They have problem in eating, similar gulping, same kind of things which we see. That the point which I want to say here is that we can use them very intelligently as a model organism to design the experiments for disease and health both. So same way, this is so at the end, to summarize what I want to say to you is that we can design the experiments by talking to Ayurvedic experts, talking to Vaidya, looking at the practice, and do the experiment, go back to them, talk to them again, show them your data, and if you get an opportunity like I have got today, you present it in front of our audience, learn from them, and this process has to go on continuously. So what I say is, this is our team, doctors, Ayurveda doctors, cognitive neuroscientists, biologists, and so many people. Then only it can happen if the transdisciplinary team is there. And the last slide is that this is what we want to do. What do we want to do? Some pieces will come from Ayurveda concepts, some pieces will come from Ayurveda practice, some pieces might come from the kind of studies we are doing, kind of study Bala is doing, some pieces will come from the public health domain, and all of that put together, the picture will emerge. And can you see the in between, the one picture, the third picture, what is that picture next to the Alice in the Wonderland? Any guess what is that picture? The second from the right? A girl? Mona Lisa? Kind of a girl, right? Second from the right? So how do we know that's a girl? The eyes are missing, right? Nose is also missing, but we very well know that's a girl, right? Picture of a girl. Because we should not be so hell-bent upon getting every piece right. We have to understand that we have to get certain pieces, we have to put them together, and the picture will emerge. We will understand what the picture is. We have to do some informed extrapolation, and then the picture can be guessed. We can definitely understand what is happening. So it is not it will be futile exercise if we say that only once only once I get all the pieces, I will decide what that picture is. Our brain is good enough that we can actually think through, and then from the information which is coming from science and practice, we can put it together and derive our own inference by doing extrapolations, informed extrapolations. So that's what Ayurveda says, Yukti, Pavanji, Yukti, I don't know what it is called, but I think Yukti is the word which Ayurveda says. We have to apply that. And that's why the AI won't replace Vaidya. That's for sure. So we have to use AI. I also felt that initially, that even the scientists will lose their jobs, but I don't feel so, because there is a lot to do, and a lot of good information can be generated and can be offered to the community. So thank you once again for your patient listening. All of you are welcome to visit TDU. It's a very good campus. Do come and do tell us what you feel. Any comments, suggestions are open. Thank you.
Thank you, ma'am, for your wonderful insights and for generously sharing your knowledge. I'd like to welcome Dr. Pawan Kumar, chairperson of this session, and invite him to offer his expert insights and observation on this topic.
Questions for her?
If anyone is curious about any topic or any doubts on this session, you can kindly ask your questions.
Any questions? You have? So ask, please. Mic for Dr. Bala, please.
Mic?
Yes.
Yeah, you can tell.
Yes. I didn't. What does it say? Please.
So as you saw in the team, there are 2 doctors, Ayurveda doctors in the team, and that's precisely what I got when we discussed this. That's what we got, that the proportion of Brahmi Grita, the Brahmi Grita which we have made, is 1 gram of, 1 portion of Brahmi Kalka, 4 portions of ghee, and 16 portions of swaras. So you can imagine how much of bitter things we have. It also has Trivrit, it also has Vacha, little bit, and Shankupushpi. So all this is there, which is actually making the digestibility of the ghee very high. So that's why, even if we see in the plain ghee, we get varied results. The plain ghee, it's, the standard deviation, standard error is very high. Not that everybody who eats plain ghee can get lower cholesterol. But with Brahmi ghee, with bitterness being there in such a high quantity, I think that's what makes the effect of the ghee, changes the effect of the ghee also. Ghee changes effect of Brahmi changes effect of the ghee. It lowers the LDL and it actually increases the HDL little bit. So that does happen. But none of these values were statistically significant, but it is just the trend which we see. In some people, actually it goes down quite a bit. So, yes. It's a Caenorhabditis elegans is normally called as roundworm. It's a very well-known model in biology. Like we have Drosophila, you might have heard of Drosophila, the fruit fly, mice and rats. Like that, it's a very common name. And we have the genetic centers from where we get. And we have the good worm scientist community in India also. In Chennai also there are many. So that's one advantage. Again, thanks for bringing that up. We have a lot of resources. We have worms which are expressing Alzheimer's protein. We have worms which live 2 times longer. And we have worms which live half the lifespan. So we have many things which we can understand using the biological nature of that particular strain. It's called Caenorhabditis elegans. It's a soil-borne nematode, non-pathogenic. So it's a safe.
centers we can get it, that one.
Yeah. It's not difficult to grow the worms, but if somebody is interested, they can come to our lab anytime, and our lab works on it. So, and we have some in Chennai also.
How you are feeded that Brahmic rhythm to that C. elegans worms?
So the worms are grown in Petri plate, and they normally eat bacteria. So we mix the ghee with bacteria, and they eat that with bacteria.
Thank you.
How the intellectual and cognitive functions were measured in worm skin?
Worms are definitely not as intelligent as we are. That's a good thing actually. Otherwise, we cannot do experiments with worms. But they have memory.
Okay.
So what happens, jokes apart, but what happens is that as they grow in the soil and they don't have eyes, so whatever they want to eat food, they smell it well and then go towards the food. So their sense of smell is very good. The chemo sensation we call it. And then they remember that this smell means this smell means danger, bad bacteria, this smell means good bacteria, and we can use that as a associative memory. The associative memory?
Yes.
The associative memory we can check in the worms. And majorly other things are, their sense of smell also will go down with age. So all that we can measure. At the behavioral level, at the genes level also. We know which are the genes responsible for this. So we can do that.
That is, after the study, that is intervention of Brahmic rhythm, the worms we used for intervention can be go for phenotyping and identification of the genes materials. From that we can conclude.
Yes.
Like that, ma'am?
Yes. So we can look at the phenotype, we can look at the gene expression, and then we can also look at the protein expression. So we can do various things.
Thank you, ma'am. Highly informative session.
Thank you.
And knowledge provoking, and having a research initiation through the young generations. Thanks for the opportunity.
Thank you.
I have a strong temptation to subscribe your statement as the concluding remarks, because it was indeed a very enlightening session. Thank you very much, ma'am. I thought that I was reading paper in JIM, which is I acknowledge TDU collaboration in that publication also. So very interesting thing. And Balaji, that worm intelligence, natural worm intelligence is akin to artificial intelligence. So good things. There are so many areas, but take home message for all of us, like an old man like me, is that shift to Brahmi Grit. So not go, so do that. Thank you very much. I will not take too much time and hand over the mic to Dr. Srinivasanji from NIN. Two nutritionists.
Yeah, it was very nice talk, madam. As well, even I too don't know much of the information. Actually, I learned from your talk. But only thing is, actually, we have many recipes are available at country level, which needs to fulfill, and validation is required. Even though I don't know, actually, the experiments which you have done was very nice results which you mentioned. But I don't know the recipes which you fed with animals are validated or not. That is my personal thing, I don't know. But anyhow, the information is very nice. Such kind of these things, when compared to the modern medicine, Ayurveda medicine nowadays, Pawan sir already has emphasized regarding the importance of Ayurveda medicine, as well the Honorable Prime Minister. Sir is also, wherever he is going, he is particularly emphasizing the importance of Ayurveda medicines. We need to come up, especially the youngsters who are sitting in this hall, they need to take as a challenge to overcome all these things and the gaps which we are finding at community level using this Ayurveda medicines. Thank you very much, madam, for your valuable suggestion. Thank you.
Thank you, Dr. Ashwini, for having patiently answered each and every question. Dr. Pavan Kumar for the laugh that you added to the session, and Dr. Srinivasa Rao for your insights. So now we would like to invite Dr. Raman Ranjan to felicitate our speaker, Dr. Ashwini Godbole, and present the certificate and memento. As a mark of our esteem, I again invite Dr. Raman Ranjan to present a memento to the chairperson, Dr. Pavan Kumar, and co-chairperson, Dr. Srinivasa Rao, in recognition of their guidance and contribution to the session.